All right, welcome everybody to the habitat risk software tutorial. If you were here for the positives or the hazards tutorial, video is working. I will turn on my video. Hey, there I am. Hello, everybody. Anyway, if you were here for the positives or the hazards tutorial, welcome back. And if you were, just feel free to tune me out for a minute because I'll be repeating things for a couple slides. So my name is Kara Them. Some of you may have heard me talk a little bit in the CWD tools webinar, and I've been sending you all those emails from SOP4CWD for the past couple of months. Also, Brenda, you might want to mute yourself. I'm a statistician and a data engineer who started working on this project back in April, and I took over project management from Brenda in August or September, something in there. So feel free to direct questions and comments and everything to me as needed. I'm really excited to be able to help you understand our new apps. I know it's been a little while since we gave these ones to you, so hopefully you've had a chance to play around with them a little bit and familiarize yourself with them and figure out any questions you have that I can address today. If you have any questions now or as we go, please put them in the Q&A section on the webinar, and I'll answer as many as possible after I go through the app with you all. And I've got a bunch of people here to help me answer them. So as you can see, I have at least pictures of the Habitat Risk app open right now. I'm going to be using Tennessee's version since James Kelly has been generous enough to give me permission to show it to you all. Your state or province's application works exactly the same for the first tab, assuming you have precise sampling locations, but the rest are going to look very different if you don't have any CWD positives in your state. I'll show you examples as we get there. Since I'm not actually going to be in the app until the Q&A section, if you'd like to open yours and work along with me as I go, you're welcome to do so this week. But you might want to jump out if we do have questions that require me logging in later on. If, when you do try to get into the app, you find that your invite has expired, please email us at sop4CWD@cornell.edu. The invites expire after a week, so make sure you go to the link as soon as you get them to avoid forgetting. And I think that is all the introductory stuff. So let's get started. As an overview, before we really dive into the details, unlike in the previous couple of apps, maps are not broken down by county, and keys to color coding can now be found in the upper right-hand side as well as the lower left. But zooming in and out works the same as in the other apps. As well, unlike the positive and hazards apps, this one requires relatively fine-scale geographic testing data. That is, we can overlay your disease testing data onto space if you have each point represented by a latitude, longitude, UTM, or some other relatively exact location coordinate. If your data is only depicted at the county level, then no part of this app or model can help you. You just won't get one. For those of you who have recorded precise geographical locations in your testing data, then you will get a partial or a full habitat risk app. I will discuss this more later, but you will get a partial app if you have precise location data but no positives, and a full app if you have precise location data and positives. Okay, so before we dive into the testing data and how that fits into the model, let's take a look at what this base map is telling us. We are looking at a subsection of Tennessee that contains the area of CWD outbreak that we've been focusing on throughout these tutorials. That tile of translucent color, in this case purple, that is overlaying the gray road map for that area is actual geographical data for that area that has been collected by the government, universities, or their affiliates and placed in the public domain. Indeed, that same data exists for the entire US, and some data exists for Canada. But if we were to try to overlay the entire map with that data, it would take days or maybe even weeks to load. Corey Mitchell, you've probably heard her name before, downloaded all that public geospatial data for the entire Eastern US, and then wrote some slick R code to chop up all those geographical data layers into state-by-state tiles, which are then further pared down into this smaller sampling grid tile once the user starts running the habitat risk software. While it would admittedly be nice to have the entire map filled in with all that geospatial data, the reason why we've only displayed a tile of it is because it would take far too long to display. You would probably get bored and close your machine, and we'd lose you. But rest assured, if you are interested in running the software for the region just to the west of the tile, or just to the east of the tile, or anywhere in the Eastern US and Canada, Corey has that geospatial data pre-processed for immediate upload into the habitat risk behind the scenes. Even better, Corey has shared her geospatial processing code via archival in the eCommons library. While the code was developed to accommodate this habitat risk app, it is quite handy for any research that requires the paring down of massive and time-consuming publicly available geospatial data into manageable chunks. Who knew there would be an entire research project geez, that sentence, man, that we would have to develop before we could even talk about the model or the app. Anyhow, back to the app. The boundaries of this purple tile was selected for analysis because it contains both CWD cases and data that has been recorded for latitude-longitude locations. We're currently looking at an elevation map. You can see from the key that the darker blue indicates higher elevation on this map. Does elevation appear to play a role in either testing or disease positive status? We can answer all those questions because the map shows us the location of all tested deer overlaid on the elevation map, or even differentiates that overlay in testing by sex or age. I just clicked on all tested deer, and we can see the great big smudge that is all the tested deer locations in this section of Tennessee. The lack of data points outside the purple tile doesn't mean we didn't test there. It just means that those are outside of our universe for this particular model. We only care about the spatial area and all the data within the purple tile. We can see from the secondary key in the lower left-hand corner that red dots are positives and white dots are negative. Those of you who were here for the positives tutorial may remember that Tennessee has tested a lot in their outbreak area, so it's understandable that there is a lot to see here. I could also color code these separately by sex, where white is female and black is male. And then if I wanted to, I could layer on age, which makes for a fun and colorful map. Here, purple is adult, green is fawn, and blue is yearling. But there have been so many tested here that the colors overlap quite a bit. Now I have to point out that depending on the amount of data you have tested, there might be a delay in loading all your points. I'm specifically looking at you, Wisconsin. You guys have done a huge amount of testing, so your app runs slow at this point. Now, if I want to calm the map down a little bit, I can turn off all deer and pick, because I like to, fawns only. Notice that I also have to uncheck by sex and by age, because they'll stay on the old testing locations until I do. But once I've selected fawns only, I see that once again, females are white and males are gray. It's a little hard to see the white against the light blue of lower elevations on this map. We recognize that, but that problem will be lessened in some of the other habitat maps. So maybe now I want to look at a background tile of slope instead of elevation. So I can click over to the slope tab at top. I have the same options for choosing to look at all deer by sex or age, or adults, yearlings, or fawns. I'll pick fawns again because it's a little easier to see the slope map under the slightly smaller amount of tested deer. Similarly, I can switch between forest, streams, clay, open, and developed habitat maps, the way I switch to slope. For example, I'm going to look at streams with all deer tested by age here, or all tested deer by age here. But as you can see, especially in this area, it's really hard to get any sense of a pattern from this map. Testing is pretty much going on everywhere, which is good, but it makes it hard to pick out patterns. So what if I want to just look at my CWD positive deer only? Could there be a spatial pattern there? I can go back to the menu and click on maps of CWD positive deer. Here we are in the CWD positive map section. For now, the map looks exactly the same, and you can see that I have the same habitat background options as I did before. But when I click on CWD positive deer, I see the locations of the deer that have been tested, that have tested positive in this area, overlaid on that elevation map. From here, I can look for patterns. I don't think I see any, but maybe on another habitat layer. This sort of pattern recognition exploration is one version of what we statisticians call hypothesis generation. Even if we saw a pattern as plain as day, simply mapping it would not count as scientific evidence that the pattern exists. However, by visualizing your data in this type of way, you can generate hypotheses to formally test. Once we can formally test it, we can ascertain whether that pattern really does exist or if it just showed up by coincidence. Okay, so I've gone through all of them on my end, and the only one that really jumps out to me is that CWD positive deer don't seem to want to be in Metro Memphis, which is hardly surprising. Other than that, there does not seem to be much of a spatial pattern to this data, or at least not one I can detect with my naked eye. Before we go on, notice that the CWD positive map not on the CWD positive map. The dots will actually overlay atop one another rather than replacing one another. So let's say I want to look at my positives by sex. Now we can see that the white and gray overlay on the red. I can add age into that and see a sort of combination of the sex and age colors. Now, what if I'm one of those lucky states with no CWD positive deer? Then what would I see? No CWD positive points. No point would appear. For example, we are taking a brief detour to the New York habitat risk interface. We've got the same spatial layers in the tiles, the same gray roadmap background, the same key, but when I click on the CWD positive deer in the Maps of CWD positive deer tab, nothing happens. So if your map looks like that, then congrats. You have no CWD in your state. And the geographical selection that bounds the purple tile really just boils back to where you've been most testing. After all, we want something for you to look at. At the very least, you can explore whether your testing follows any spatial pattern in the previous tab. Like we've seen, we can display Cori's pre-processed geospatial data for any state or province for any agency that collected their testing data records with exact location. So everything I've just shown you is what we call the partial app. The full app has a bunch of modeling stuff on it, which I will go through next. The first tab relevant to the full app is this one, the estimated relative risk of CWD tab. If, however, you see this screen for your own state, then congrats. You don't have CWD, and so most of the rest of this tutorial is going to be purely academic. However, even if your app shows this screen now, it contains the parallel universe in the code that contains the modeling infrastructure for when your state or province gets CWD. Hopefully that never happens, but it is there lurking in the background, just in case. All right, so unfortunately you do have CWD. In this case, the estimated relative risk of CWD tab will contain a gridded out map of modeling results. The map has been automatically divided into 6 square kilometer squares, which correspond to the best guess at the average home range size of deer for this area. We have used 6 square kilometers for all of our apps, but altering home range size to any more precisely estimated size means altering one line of code. So it's very easy to do. In version 2 of the warehouse, we're actually going to let you enter your own home range size, but that's in the future. For now, your app displays 6 square kilometers. Each grid cell is auto-filled with color according to the modeled relative risk of harvesting a deer with CWD infection from that cell. The map I have displayed is the risk of harvesting adult bucks, but similar maps exist for adult does, yearling bucks, yearling does, fawn bucks, and fawn does. In fact, you can click through the tabs at top to see the segment of interest. In all of the maps, relative risks are arranged on a scale of 0 to 1, where 0 means there's a 0% chance that a harvested deer will be CWD positive, and 1 means there's a 100% chance. In the displayed map, the darker the red in the cell, the more likely you are to harvest that CWD positive buck. Some cells are grayed out, meaning that the algorithm doesn't have enough information to estimate risk in that cell, probably because you haven't harvested sufficient deer from that exact cell for the algorithm to really know what's going on there. As is always the case with maps on our apps, we can click to see the high resolution details of the data contained in any one of the cells. In this case, those pop-ups allow us to view the median and the 95% credible interval of risk for each age sex segment. As you would probably anticipate, fawn does are the least likely to come back positive, which you can see by the relative paleness of the fawn doe map. If you get confused about what's happening on this map, you can always click the "How do I interpret this" map button at the bottom of the page, which gives us a reminder of what the cells are telling us. Now, don't be deceived by the simplicity of these maps. Each one of these maps required up to 14 days of runtime, as well as no fewer than 7,600 lines of code. And that doesn't even include Cori's geospatial preprocessing. We show you the map before all of the details about the underlying model, so you can see up front the results and hopefully the usefulness of this model. But there are a lot of steps in the model that were necessary to get here, and we're going to go through these now. So buckle up. The model selection page lets us get into the actual nuts and bolts of the statistics happening behind the scenes with this software. Basically, what's happening is that we're running 25 possible Bayesian hierarchical models to estimate the relative risk that a grid cell will harbor a CWD positive deer based on the geospatial habitat conditions that we saw in all of those publicly available map layers displayed on the first couple tabs, and additional explanatory variables such as the sex, male or female, and age, fawn, yearling, adult, plus the regional and local variation. The details of those 25 candidate models are listed out on this page. Those candidate models are the same for everybody. While running each model, the data are fitted to the structure of that specific equation. The performance evaluation of the model is conducted, and the fitness of any given model are compared to the 24 other models. The comparison is a statistic metric that we call the DIC. The model with the lowest DIC rating is chosen as the winning model for our data and gets placed in the Hall of Fame. That is, it appears at the top of this model and becomes a standard by which all other models are compared. Its Hall of Fame status also means that it is the model that is used to create all those statistical computations and generate that risk map on the previous page. We can scroll down a little to see what the variable names in these model formulas represent. I know there's a lot of words and they're small here, so I highly recommend taking the time to go through it on your own app to see what's happening with those candidate models. If you think this is interesting stuff and you haven't already, you might consider getting an MS or even a PhD in stats, because we need you out in the field. Alternatively, hire a statistician and pick their brain a lot. If I scroll down even further, I can see the results of the candidate model selection process. Each model is listed in order by smallest DIC to largest. Note that we actually did a little bit of expansion to the Tennessee grid to get a larger area in the model, so I've actually pivoted over to showing you Pennsylvania's models here. I'll zoom in a little so we can see better. As we can see, Model 20 was the best model for the area selected in Pennsylvania. It came out on top of the list with the lowest DIC rating. Here's a reminder of what the mathematical formula for Model 20 is. But having Model 20 come out on top is great. That means that age, sex, and CAR, or the local variation not accounted for by the other spatial variables, are the only significant factors in predicting home ranges that produce CWD positive deer. As a side note, Model 20 seems to come out on top in most states for which we've run the habitat risk model in SOP4CWD. That is actually pretty cool, because having that result show up in all sorts of places might speak to the intrinsic properties of the disease. To add to that curious result, Model 20 is the one that doesn't actually use any habitat characteristics. So that's interesting. We'll be exploring this idea some more. The same pattern with Model 20 is also what allowed us to create that larger grid for Tennessee that I mentioned. Because it's so pared down, we could run a much larger geographical area at once without our computers exploding. If you haven't already picked up on that theme, then you will. This modeling process requires massive computational power, memory, and runtime. As always, if you're confused, there's a helpful "How do I interpret these comparisons" button at the bottom of the page, which will give you more information about how the top model was selected. But remember how I just gave you a refresher on Model 20? Well, it turns out I didn't really need to, because the best model is explained in more detail on the next tab, fittingly named the best model. I'm going back to Tennessee here because now this tab looks just like the rest of yours again. The first thing that you will see is the best model in mathematical notation. In this case, Model 20, because it is the boss, written out with symbolic coefficient values. The next line shows you that model with the coefficient values that have been estimated using that 14 days of runtime. We'll also show you what the median coefficient values for all the cells are for each of those variables, along with a key to explain what those crazy notations are. We give you a breakdown of each coefficient value as well, so you can see various statistics in them. If you're confused about all this crazy statistical math stuff, click on "How do I interpret these values?" This gives you a little more behind the scenes look at what's happening here. But if you're really lost, what might comfort you most is that, like the previous screen, although I didn't mention it, what you need to do at the very bottom there tells you nothing. You're good. You must proceed by doing nothing. The computer did it for you. All this stuff just exists to help you understand what's happening if you want to know more about the nuts and bolts of the model. For the biometricians out there like Brenda and I, this is very helpful stuff. But for the rest of the world, the relative risks might be all you need to care about for now. One thing, however, that you should be aware of is that there are some diagnostics that you should check out or get someone else to check out. Either way, Diagnostics exist to ensure that the automated computer didn't just happily run the script right off a cliff. In other words, we want to make sure that the computations met the statistical milestones that it needed to in order to produce valid results. If I dismiss the help text and scroll down on this page, I see a number of diagnostic radial buttons. These diagnostic tables and plots should be checked to make sure that the model ran the way we expected it to and converged the way we wanted it to. If it didn't, then all of those previous results don't actually mean anything, and worse, maybe misleading. For the apps online right now, we've been checking the diagnostics for you, but you should get in the habit of doing so yourself. Because once you're running the models on your own through the warehouse, that oversight on our end isn't going to be as readily available. Plus, it's always good to check twice anyway. So let's just take a quick tour through these diagnostics so you have an idea what you're looking for. Note that for each one, you can click on the tab that tells you how to interpret it. So starting at the top, Goecki's convergence diagnostic. If I click, how do I interpret the Goecki's convergence diagnostic? I see some information about the diagnostic, as well as what I should look for. In this case, I should check to see that basically all of the numbers fall between negative 1.96 and positive 1.96. In a nutshell, the computer is estimating the statistical distribution of each parameter. If all the numbers fall between negative 1.96 and positive 1.96, then you can be 95% confident that distribution is settled to its final value, which is what we want to have happened, and is no longer moving around, which is what we don't want to see. Gelman and Rubin's convergence diagnostic is used to ascertain whether a Markov chain is ergodic. In other words, whether each chain has forgotten its initial values. Also, every time I read this slide, I laugh because Brenda gave me a nice phonetic spelling of ergodic, just in case I forgot. We want the chain to forget its initial values. If the potential scale reduction factor for parameter is at 1, then you are golden. If you get confused, you can always click the help text and read the snippet on the diagnostic. Similarly, the Heidelberger and Welch's convergency diagnostic is another way to verify that the chain has arisen from a stationary distribution. If you get this diagnostic and it has passed, then everything is good. Rafferty and Lewis's diagnostic ensures that the thing we call the burn-in is sufficient. Now that sounds like some sort of firefighting joke, but it's actually not. Recall that we want the chain to forget its initial conditions. The burn-in is simply the number of iterations that it takes to do that. You can check that you have achieved this diagnostic by verifying that we have sufficient samples when it comes to the effective sample size, which I will discuss next. The effective sample size is the number of samples that bring independent pieces of information to the problem. The classic way to teach the difference between a sample size and an effective sample size is to think of family trees when you wish to explore genetics. A mom, say, brings one independent packet of information regarding the family's genes. Her daughter's genes overlap with her genes by about a half. So the daughter's genes only provide about a half of additional independent information when pooled with the genes of the mom. So despite having 2 people in the tree, or a population size of 2, we have an overlap in information. So we only have an effective population size of 1.5. The more overlap 2 or more data points have with one another, the less independent pieces of information are brought into the problem. Effective sample size is the smaller number. So to check this diagnostic, verify that the effective size exceeds the minimum information content necessary for the Rafferty diagnostic, the one we just discussed in the previous slide. We also have the trace plots. These trace plots show 2 things for each parameter. First, they show the thing we like to call the hairy caterpillar because it looks sort of like a hairy caterpillar, that teal blob to the left. It should look random. If it does, it means that the sampler under the hood is doing its job. So that's great. The second thing these trace plots show is the distribution of each parameter. Recall that we wanted to make sure it was stationary? Well, not only stationary, we want to make sure it has some sort of distribution. That normal curve over on the right shows those distributions. From those, we get the estimates for each of the graduated cell colors in our risk maps. And like always, you can click the help box to get a quick reminder of what these things are meant to do. Finally, we have the correlations plot. These just ensure that the pieces of information used to estimate each parameter aren't correlated. We don't want them to be, so a triangle of bright green is our goal. Now, these are kind of hard to interpret in some cases, including this case, because the whole thing probably just looks black to you. That is because we have so many cells in our sampling grid of deer that each cell in a pairwise correlation matrix is smaller than a pixel. If you were to select a smaller sampling grid or have a larger computer screen, this black triangle will expand and you will see the colors you are trying to see. Remember, green equals good, and that's basically what it comes down to. And as always, you can click the help button to get additional information. And that's all the diagnostics. We got through them. That was a lot of stats. But as you can see, this is why these models are so challenging to use in practice. They require some pretty heavy simultaneous understanding of several highly technical mathematical concepts. This is about as high-level stats as it gets. So let's move on from this very long and statistically dense page and look at the Explore the Maps page tab. Here I find what looks like the exact same page as I saw earlier in the relative risk maps. But the difference is if I scroll down just a little, I have the option of downloading the risk map. If I click download this map, I will download an HTML leaflet file that contains all the information found on this page of the app. The beauty of this feature is that you can scroll, pan, or zoom the map, and the exact map that you are looking at will be the one that gets downloaded. At the top of this page, you will see that you can do this for any of the 6 age sex segments. Last, but certainly not least, we have the tutorial page. Now this one is a bit longer than our previous tutorials because, as you've heard, there's a lot to unpack here. But it helps explain what each page does and when you might or might not see it, depending on your state's CWD status and location information. And that's it. Thank you so much for listening. Hopefully this has been helpful in deciphering what is going on with this software. Again, please ask any questions in the Q&A rather than in chat so we can keep track of them. This week, I have Kristen, Brenda, Nick, and David, who created the underlying model, here to help me answer your questions. So looks like we've got one question in Q&A that I haven't read yet. All right, it says maybe semantics, but it seems like what you're calling relative risk is actually just risk. Probability of infection rather than probability of infection relative to a reference, or relative to a reference, referring to the map. Brenda, you have an answer. It looks like you're nodding because I'm still trying to compute the question in my head.
Yeah, we're just using the word relative simply because it can be either interpreted as the probability or the odds of infection. And so when it's odds, then it's the relative risk.
There you go. Semantics indeed, but
Yeah.
good question. Answered. Anyone else have a question? I've got all these experts here. You got to pick their brains, you guys.
So an interesting thing that is happening behind the scenes that I'll expand on a little bit when Kara said that we have 7,600 lines of code. So we've got the 25 different potential models, and depending on the model that's selected for each bit of data, all of the interface that you see depends on the structure of the model that was selected. And so that's part of the reason why you have 7,600 lines of code, is because you have 25 potential interfaces. And there's a decision tree based on which model is selected. And so all of this happens with one click, which is pretty exciting that we were able to make that happen.
Yeah, good work, all you modeling people.
There's a lot of coding going on behind the scenes of this one, but there's a lot of flexibility in this one also. As Kara mentioned, there's the ability to change the home range size. There's also the ability to change the selection of layers. Right now we've got streams, elevation, forest, that kind of thing. But provided those layers exist, you can swap them out. And then if you wanted to even add or change the layers that you have in those potential models, you can do that. And so we've also published this habitat risk software to the eCommons website as well, and shared it under an MIT license, which means that if you know how to code R and you're interested in modifying this for a different area, for a different disease, for anything you want really, you can go in there and download it and modify it. And then provided you cite at source, you can use it for a different application.
freedom. You have freedom. I'm not getting any more questions though, even with all my experts. I probably broke all their brains with the diagnostics.
Yeah,
I broke my brain with the diagnostics.
Me too. Yeah.
So as always, if you guys have thoughts or ideas or feedback on the apps, please let us know so that we can make sure to incorporate those into version 2, because I'm in the middle of working on it. So want to get it all right. And for next week, we'll be doing the optimization app, which I don't think anyone has access to. Is that correct, Brenda?
Not yet. Well,
Not yet.
Tennessee and PA do, but not everyone.
Okay.
But yeah.
So we'll do a tutorial on it and we'll be getting it out to you hopefully fairly soon. But that way, at least you'll know what's going on when you do get it. So anyone else have anything they want to add before we call it?
Thanks, Kara.
Thank you. All right. Well, thank you everybody for coming. It was great to have you. And I hope you have wonderful weeks and we'll see you next week for optimization.